to be giving this talk in the Bio-Inspired Soft Matter series at Brandeis. So I was actually at Brandeis, I think, gosh, now 6 or 7 years ago. And at that time, I think I spoke almost exclusively about the cytoskeleton. But today I'm going to be mostly talking about some ongoing work in my group on more like tissue mechanobiology and collective behavior that gives rise to this tissue mechanobiology. So, I think we tested and everything was working. Okay, yes. So I think I don't have to convince this audience that, you know, mechanics is really important for cell and tissue form and function, but still I wanted to showcase that a little bit. So, you know, so first there is this movie, which I guess all of you have seen, maybe even hundreds of times. It's got like about 4 or 5 million views on YouTube. So this is a white blood cell, which is relentlessly chasing a bacteria. And, you know, as you can see, and the mechanics of this cell is really important in the cell being able to sort of pursue these bacteria to generate forces which will help the cell to move, to push the red blood cells out of its way, and doing all of this while maintaining its mechanical integrity and at the same time changing shape that allows this white blood cell to sort of move through narrow regions, right? So the mechanics of the cell is really important for it to be able to do this. And the second example I wanted to showcase is this one from this paper now over a decade ago from UPenn, which was basically looking at pluripotent stem cells which have been kept on substrates of different stiffnesses. And what this paper found is that if you take one of these pluripotent stem cells and put it on a substrate which is very, very soft, then the stem cell later on differentiates to become a nerve cell, while if you put it in a substrate which is very stiff, it will differentiate and become a bone cell. So the stiffness, the mechanical stiffness of the substrate sort of helps to guide the differentiation of the cell to decide, you know, helps the cell to decide its fate, right? So then one can ask, what physical principles, rules, and mechanisms are helping to guide, you know, living systems in you know, guide the form and function of living systems and help them sense and respond and adapt to mechanical stimuli in their surroundings, as well as generate, transmit, and exert forces, you know, on their surroundings, right? So these are some of the questions that, you know, we would want to understand, right? If you want to understand what is sort of the basic, what underlines the basis of this kind of mechanobiology. So the mechanobiology of cells mainly arises from the cytoskeleton of the cell. So like we have our skeleton, cells also have their skeleton, which is the cytoskeleton, but it's a much more living, breathing structure. And I like to think of the cytoskeleton as consisting of two major players, the shapers, which are basically these biopolymer networks cross-linked with different types of cross-linkers and so on, which are responsible for the shape and the mechanical response of these cells. And then there are the molecular motors, which are responsible for force generation, contractility, and transport of you know, entities within the cell. And I like to think of them as movers, right? So the cytoskeleton is then mainly what's giving rise to the mechanobiology of cells. Now, if you want to think of the mechanobiology of tissues, well, let's start by looking at this image of a tissue. And you can see that this looks really, really complicated. So you have these cells which are embedded in this very heterogeneous network-like extracellular matrix, right? So this looks very complex. So I am a theorist, and we have sort of a spherical cow picture of every situation. So this is my theorist spherical cow picture of this, you know, tissue. So it has, you know, these extracellular matrix fibers, which are like a network, and it has these cells. But even this picture is very complicated because, you know, the cells are not just, you know, just some passive entities. These are active. They have their own interaction between themselves. There can be interaction between the cells and the ECM and feedback and so on. So, and then, you know, these systems can also be quite heterogeneous. So this is still a hard problem. And the way I like to deal with, or I like to address this problem, is by breaking it up into modules, right? we can look at what's going on at the level of individual cells. We can look at what is going on in tissues, which are mainly made up of this network-like extracellular matrix. And we can look at maybe tissues or tissue-like systems, which is mainly governed by what's going on with, a collection of cells, right? And so that's how I think one can make this problem a little bit more tractable. And in my group, actually, we are looking at mechanics and dynamics at the level of cells, as well as at the level of tissues, and looking at a number of problems. And today I'm going to talk about mechanical response of tissues, but I will focus on two different types of systems. I will focus on tissues that are mainly made up of this extracellular matrix, and sort of our model system here is cartilage tissue. And then I will talk about tissues or tissue-like systems that are mainly made up of cells, and I'm going to talk about a co-culture of two cell types, the breast epithelial cell type MCF10A and the breast cancer cell line MDMB231. And I guess I also wanted to mention that if you guys have any questions, please, if you have any clarifying questions, at least, please ask me during the talk. Do not wait until the end of the talk. Okay, so let's start by looking at this tissue, whose mechanics is mainly coming from this network-like extracellular matrix. And the tissue in point, that we are focusing on is articular cartilage. So cartilage is a very thin tissue. It's only a few millimeters thick, and it's present in our joints, in our knees, and our elbows, and so on and so forth. And this tissue, although it's very, very thin, it can help us bear loads that are up to 10 times our body's weight, right? And the other thing is that, like I told you, it's mainly made up of this extracellular matrix. Now, extracellular matrix has to come from somewhere, and it's basically made by cells, right? So cells are sort of, making or secreting this ECM that this tissue is made up of. Now, once we become adults, our cartilage tissue starts having not very many cells. It has got very few cells left. So whatever cartilage or whatever articular cartilage we have when we become adults, we are going to have the same cartilage for the next 60, 70, 80 years of our life. We are not going to be making any new cartilage. So then during all of these decades of our life, however, as we keep on, running, jogging, doing our daily activities even, our joints and our cartilage tissue is going to undergo several 100 million loading cycles, right? So then this tissue has to last us our whole lifetime, despite having this very limited regenerative capacity, which means that, it has to be really tough and strong and crack resistant and adaptive and all of these things at the same time. And we haven't really come up with any synthetic materials so far which have the same functionalities as cartilage. So then you can ask, well, what kind of physical principles and rules are responsible for the mechanical structure function properties of cartilage? And, can we also predict, when will we start seeing tears or fractures in cartilage, right? So these are some of the questions we want to understand. And I guess I should also tell you a little bit about the composition of the tissue. So it's mainly made up of water, and then there is network of collagen fibers, which is quite stiff. And then there is a much more flexible component, which is coming from proteoglycans and hyaluronic acid. So you can think of it as sort of a more floppy, flexible, networky thing. And the work I'm going to describe today consists of experiments that were done that are being done in my collaborator, Itai Cohen and Larry Bonassier's lab at Cornell. And the theory that I'm going to talk about was worked on by my students, Andrew Sinderman, Leo Sutter, and Piancy Lewin. So we want to understand the mechanical structure function properties of cartilage tissue. And this tissue is a three-dimensional tissue, right? So the first thing we wanted to understand is how does the mechanical response of the tissue change as a function of tissue depth. So
can I ask you a quick clarifying question?
Yes.
All right, thanks. So this is all about a small tissue that's on top of the bone that's like in our knee?
This is in the joint, right? So in the joint, between two bones, you have this tissue. So in this cartoon, if you look at this, right? So you have a joint, the red part So the colors in this figure, basically the red ones are samples coming from this patellofemoral groove, and the blue curves are coming from sample which is in the tibial plateau, right? And in this x-axis, when I say 0, 0 means you're close to the joint, and the 1600 microns means that you're close to the bone, right? So,
Okay,
so
that makes
that's
sense.
what's the two ends of the x-axis mean.
All right, thank you.
Okay. Okay, so then the first we wanted to look at the shear response of this tissue as a function of tissue depth. And why are we looking at shear? Because if we want to, understand tearing and those kinds of properties in a tissue, it would be interesting to look at how they respond to shear. So, and you can see that for, when you are very, very close to the surface of the joint, the shear modulus here is very small. And then as you go deeper and deeper and get closer to the bone, the shear modulus actually increases quite a bit. Also note that the x-axis is on linear scale and the y-axis is on log scale, right? So in the x-axis, as you go from, let's say, here, from 0 to about 800 in microns, the shear modulus increases by about 3 orders of magnitude, right? So we wanted to understand why is the shear modulus or the shear stiffness of the tissue increase by several orders of magnitude as we are going deeper in the tissue, or rather than why, how does it happen, right? So if you have looked at cartoons of cartilage in tissue engineering books, these are the kind of cartoons you would have seen. So these fibers are supposed to stand for collagen, and these are the few cells that are left in cartilage that I was talking about, right? So you can see that close to the surface, close to the joint, the collagen fibers are all aligned, parallel to each other, but they're also aligned parallel to the surface, right? So if I shear it, the energy cost of shearing is going to be very small, and this can explain this kind of small shear modulus. But close to the bone, once again, the fibers are aligned parallel to each other, but now they're perpendicular to the surface. So now if I try to shear it, the cost of shearing is going to be quite large, and that can explain this larger shear modulus. So that was our initial hypothesis, that this depth-dependent mechanical response of the tissue is mainly arising from the organization of collagen fibers in the tissue, and how does that depend on depth. But this is a hypothesis, and we can test this hypothesis by looking at how the shear stiffness sort of correlates with different measures of orientational order for these fibers. And I should also mention that these experiments were done at very small shears. So the shear strain that's applied here is less than 5%. And what we found is that, well, collagen organization might be there, but for these kinds of small applied shears, there is no correlation between the shear modulus and either the polarization index or the average orientation of fibers. So the polarization, the average polarization index, is actually a measure of orientational order in these fibers, the collagen fibers. So if the collagen fibers are all aligned roughly in the same direction, which is happening at the top and the bottom, then the polarization index will be 1. And when the collagen fibers are sort of isotropically, distributed, then the polarization index will be 0. But you can see that in this graph, there is absolutely no trend, right? And Itai and his student also looked at the shear modulus as a function of average orientation of the fibers. And once again, there was no correlation between the shear modulus and the average orientation of fibers. So even though there may be some orientational ordering in the collagen fibers in this tissue, that is not what's giving rise to this shear response. So then what else could be going on? So the next thing that my collaborators did is they looked at this shear modulus, but now as a function of the volume fraction of collagen in the system. And once again, the collagen volume fraction here on the x-axis is plotted in log scale, and the shear modulus is plotted in linear scale, and the shear modulus is plotted in log scale. And what we found is that for small collagen volume fraction, the shear modulus is quite small. And then as you increase the volume fraction, once again, it increases. And as you increase the volume fraction, let's say from 15% to 25%, this modulus, the shear stiffness, increases by, again, almost 3 orders of magnitude, right? So then you can ask, well, how can we explain this, right? What could be driving this? Now we know this from, our and others' previous studies of in vitro biopolymer networks, that, if you see that you are changing the volume fraction or the density of filamentous material in your system by a small amount, and your modulus changes by a huge amount, this is basically indicative of a mechanical phase transition, right? And in these kinds of disordered biopolymer networks, this mainly often arises because of this phase transition, which we call the rigidity percolation phase transition. So let's see if that is what is happening here. But before that, let me briefly tell you sort of what this rigidity percolation approach basically says. So in this approach, we model these biopolymer networks as disordered networks made up of semi-flexible filaments. So each of these filaments can stretch and bend, which will cost some energy. And if your network is very, very dense, if you have a large amount of filamentous material, you can get these large spanning clusters in your system. So the red structures here are basically showing connected spanning clusters. So if you have a system like this, if you try to shear it, it's going to resist that shear, and you're going to get a finite shear modulus. On the other hand, if your network is very, very sparse and you get this really small connected spanning clusters, then if you try to shear it, it's not going to be able to sort of resist the shear, and you will get a shear modulus of 0. So then you can think of it like if you're sort of tuning the amount of filamentous material or the volume fraction of filamentous material in your system, then there will be a critical sort of a volume fraction where the system goes from being non-rigid to being rigid, right? And that is basically this rigidity percolation threshold, and this phase transition is essentially this rigidity percolation phase transition.
Moumita, can I ask a question?
Yes.
So it can't just be the density, it has to be the connectivity of the network.
Yeah,
right?
yeah.
Because
so it has
you're
to
just
be,
doing
when I
Maxwell
say
function.
density, I mean here bond occupational probability. So that, you can think of it in terms of how well the network is cross-linked, right?
Yeah,
and
right.
here, because, in this model, the amount of material that you have is sort of tied to how well they're cross-linked, right? You can decouple those things, but the way the model is constructed, the more filaments or fibers you have, the more connected they are.
But if I had them like all aligned and not cross-linked, I could have the same density, but I wouldn't have this percolation.
Yeah, so you will need to have some kind of interaction, right? You either need to have really rigid cross-links, or at least you will need to have some kind of strong entanglement or something, right? Otherwise it will not. Otherwise you can have a lot of filamentous material, but they will not resist deformation. You're correct. So this was then our next idea. Okay, let's see if this is what is happening. And so then we went and constructed this model, but again, the tissue that we are looking at is a 3-dimensional tissue, right? So and it's not just a 3-dimensional tissue. The volume fraction of collagen also changes as a function of tissue depth, right? So how do we model that? So what we did is we basically said, well, you can sort of make, slices of the tissue at different tissue depths, and you can measure the volume fraction of collagen versus all the other stuff that's in the tissue at these different depths, right? And corresponding to these different depths of the tissue, you can sort of construct 2-dimensional systems where you have a 2D collagen network, which is a stiff semi-flexible network, and all this other stuff, which is hyaluronic acid, proteoglycans, etc., etc., that is sort of continuum elastic background that this network is embedded in. And also I should mention that this tissue does have a lot of water, but because I am focusing completely on the mechanics, I am neglecting any water whatsoever in the tissue, right? So then, and we are looking at what's going on when the applied strain is really small. It's less than 5%. So we can write down the deformation energy that's appropriate for small deformations in that system. So we can write down a deformation energy that has a component coming from, the stretching of the fibers, a component that's coming from the bending of the fibers. And because these fibers are embedded in an elastic background gel, and this background gel will resist large deformations, there is also another component coming from any resistance of this background gel to transverse displacement of the fibers, right? So in the small deformation limit, these are the contributions to our deformation energy. And once we have written down this deformation energy, then we can apply a shear deformation to our network, and we can do a mean field like theory. So we can do an effective medium theory, for example, or we can do a computational energy minimization, and we can find out what is the shear modulus for this tissue as a function of the volume fraction of this collagenous material. So essentially in the theory, we find out, what is this shear modulus as a function of the occupation and probability of bonds in this lattice-based network. And then we can sort of map that to the volume fraction of collagen in the system. And so the prediction of the model is shown with these dark symbols, and the experimental data is shown with these gray symbols here. And once again, in this other graph where we are showing the shear modulus, the predictions of the model are in the colored dark symbols, and the gray symbols are basically showing the experimental data. So what we see here is that even with this very toy model, the predictions of the model sort of do explain reasonably well what's going on in the experiments. So the dashed line, basically here, the blue dashed line shows what's going on with this network. I change the volume fraction of collagenous material, of collagen filaments in this network. But then there is this other line that I have added here. And the reason this is coming from, so there is a baseline response of the background gel to the total response of the tissue. And any filamentous material that has broken off from the network gets added to the background gel. Nothing is leaving the system. So as the volume fraction of the network decreases, the amount of material in the background gel goes up a little bit. That's why you see this uptick in the curve. And together, this is sort of the response we get from the network plus the background gel. And then we can map sort of the volume fraction of collagen to the depth of the tissue, because we know from experiments at given depths what is the volume fraction of collagen. And then we can recreate then the depth dependent shear response of the tissue from that. So even with this very, very toy model, this idea seems to explain the fact that the shear response of the tissue is changing by several orders of magnitude. And that's the reason that's happening is because as we change the depth of the tissue, the amount of collagen in the tissue also changes, and that sort of basically then helps to sort of trigger this percolation kind of behavior, right? So the tissue is very close to this rigidity percolation, and by changing the depth of the tissue by a little amount, we change the volume fraction of collagen also by a little amount, but that turning the knob by a little amount is big enough to sort of cause this kind of rigidity percolation behavior. But at the same time, the entire system does not fall apart because of this reinforcement coming from the background gel, which is made up of aggrecan and hyaluronic acid and other things, right? So the mechanical response of the tissue then can be understood by using these ideas of rigidity percolation. So before I go on, do you guys have any questions? This may be a good time to take a few questions if there are any. All right, so, okay, so we talked about the mechanical response of tissue at small strains, right? But we also want to understand what will happen to this cartilage tissue when it comes under large deformations or even moderate deformations, right? So for example, let's say you are jogging or exercising or are doing your favorite, activity, and you get injured, right? Can we make predictions that these small injuries that might lead to small tears, that these tears can then sort of lead to all out of fracture of the tissue, or when that will happen and when that will not happen, right? So fracture mechanics, overall is a very mature, well-developed field, right? For example, many of us are aware of this very well-known criterion called Griffith criterion for brittle fracture, which tells us, based on, the material and, a characteristic critical load or critical crack length for the system, for the material, we can predict when a crack will grow and lead to fracture, and when it will not grow and when it will heal. Unfortunately, for tissues, we don't have a similar framework because these are highly heterogeneous, composite, disordered materials, right? So our goal is to come up with a predictive framework that can make similar predictions for, soft tissues like cartilage. And on the right-hand side here, I'm showing you an experimental image of what crack growth looks like in cartilage. So the way people study cracks in tissues, in joints or ligaments or tissues is basically you will make a small notch in the tissue. Then you'll apply sort of a tension along the ends. And then we want to see how this crack or this notch is going to grow. And you can see that in this case, there is the crack sort of opens up, but it's not like a brittle fracture here, right? And the end on the other hand, on the other side, sort of pulls up a little bit. So we want to come up with, or we want to extend our model to see if we can explain this kind of crack propagation properties in cartilage tissue. So we first wanted to try the model that I just described to you, where I model the collagen as a semi-flexible or stiff network, and everything else is a continuum soft elastic background gel, right? And I make a notch in this network, and then I apply tensile stress along the two ends. And then I increase the amount of strain. I apply tensile strain along the two ends, and I increase the strain along the ends by 2%, 2%, 2%, and see what happens, right? So at 10%, this is what the system looks like. At 20%, you can see that the notch has grown, and you can see there are these other cracks that have appeared all over the system. At 30%, the notch has grown quite a bit, and at 33%, everything has now fallen apart. The tissue has ruptured into two parts. I should also mention that when I try to calculate this, now I'm taking into account large deformations, and I'm allowing bonds to break and to buckle. And these bonds will break when they have been stretched to more than 120% of their initial length, and they will buckle if they have been compressed to more than 95% of their initial length. The other thing that we are assuming that if a bond is broken, then it's broken forever. However, if a bond is buckled, then if you take the compression away, then this buckled bond can come back into circulation. Okay, so now with this model, so we can see the crack growing and leading to fracture, but this fracture looks very much like brittle fracture. This doesn't really look like what crack growth looks like in experiments on cartilage tissue, right? So my first model, although it explains the mechanical stiffness, shear stiffness of cartilage as a function of tissue depth, it doesn't really explain crack growth in cartilage very well. So we need to improvise, we need to sort of change our model, right? So what could we be missing? So then we thought maybe what's missing in our first model is that we thought of aggregate and hyaluronic acid and everything as a continuum elastic background gel, but that's really not true, right? So in reality, they are like another sort of softer, much softer, floppier, networky structure. So instead of now having one stiff network of collagen in a background gel, we have sort of a composite or a double network, right? Where the stiff network is basically, again, the collagen network, and the softer, more flexible network is basically representing hyaluronic acid and proteoglycans together, right? And the two networks interact with each other, not at the nodes, but across, at the, basically, there is weak interaction between the two, between the bonds in the two networks at their midpoints. And we can write down the deformation energy for each of the fiber networks. The stiff network, again, has contributions from stretching and bending of fibers. And also, we still have a background gel, but now the background gel is everything else, not collagen, not proteoglycans, not hyaluronic acid, the remaining stuff. So the stiff network has contribution, again, from the stretching of the fibers, the bending of the fibers, and the resistance of the background gel to deformation of the fibers. The flexible network does not have any bending energy because the filaments are very, very floppy. It doesn't cost any energy to bend them. So now there is only cost of stretching these fibers. And then again, there is this background gel resistance, and we have an interaction between the two networks, right? So now with this deformation energy, we can again create a notch in our initial network and then strain the network more and more and more, and then see what happens. And so now at 10%, this is what the notch looks like. Then we go to 30%, then we go to 50%, and then things fall apart at 52%. So there are two important differences between this system and the previous network. One is that here the fracture looks much more like what you have seen, what we have seen, these examples from experiments, right? So this is more like ductile fracture. It's not like brittle fracture anymore. And also you can see that the strain at which everything falls apart, which is basically the ultimate tensile threshold, that's about 20% larger than this case, right? So you might say that, well, I'm comparing apples and oranges because in one case, I am looking at a single network. Here I'm looking at a double network. How can I even compare the two? So the way we are able to compare this is that the stiffness of the collagen fiber network is always kept the same, but the stiffness of the background gel in this case is sort of equivalent sort of to the shear modulus, or the stiffness of the second network plus the background gel. So we adjusted our parameters for the second network so that it will mimic what was just the background gel in the first network. Now that's the second network, the flexible network plus the background gel, right? So then why is this happening, right? Why is the ultimate tensile threshold larger? And why is the fracture not like brittle fracture anymore? So what's essentially happening here is the following. So earlier, when you just had a stiff network, if you have concentration of large stresses in the network, the only way to relax that stress is to create tears or cracks in the network, and then the network will start rupturing pretty quickly, right? And you will get this brittle fracture-like behavior. However, if you have now a second interconnected network that allows these stresses to be distributed over larger areas, that's one thing. The second thing is that the second network is also very, very floppy and quite flexible and sparse. And it can also allow sort of some of the accumulated stresses to then just sort of, relax or dissipate into the system, right? And by doing this, the second, this double network can now have a much larger ultimate tensile threshold. And also we get this ductile-like fracture when we have this double network. And again, this seems qualitatively also very similar to what was seen in experiments. You see this kind of blunting of the crack, and the bottom also pulls up like this, right? So the next question is, okay, so if we want to make a double network with a stiff network and a flexible network, what kind of composition do we want? What kind of, densities, fiber densities do we want, or connectivities do we want for these two fibers or two networks to have, so that we get a material which is both, sort of strong, so which has a large Young's modulus, and at the same time is tough, which means that it's crack resistant and doesn't really fall apart very easily, right? And to do that, we looked at the Young's modulus as a function of applied strain, and we also looked at the stress as a function of strain, right? And what we did is the things we are varying are these two bond occupation probabilities for the stiff network and the flexible network. So P1 is the occupation probability of bonds in the stiff network. P2 is the occupation probability of bonds in the flexible network. So what we find is that if we have a very well-connected, dense, stiff network, then of course we can get a very large Young's modulus. However, that network is also very brittle, and it will start rupturing at very small strains, right? Now, one of the ways we could make this network have large Young's moduli, but not really fall apart as easily, is by keeping sort of the bond occupation probability of the stiff network large, but maybe not so large that makes it brittle. And at the same time, maybe making the second network, which is helping to sort of distribute the stresses and relax the stresses, sparse, right? But again, not too sparse that the second network doesn't percolate at all. And this is also sort of reflected in the stress strain graphs, right? So in the stress strain graph, if we have really large values of P1 and P2, we will get really large maximum stresses before the system fractures, but it will also fracture at really small strains. If we want the system to not fracture at small strains, then essentially, we would want to decrease P1 but maybe still keep it large enough so that you still have, at least a moderate amount of rigidity, and at the same time decrease P2 so that, we make this, much more sort of ductile-like or much more continuous, so that if you're looking at where does, the network completely fall apart, tear apart, that happens at very large strains, right? So we would want to have a material where, our stiff network is dense, but not so dense that the, system ends up becoming brittle. And our flexible network is sparse and floppy, but not too sparse that, we don't get the stresses to percolate at all, right? So this is sort of the summary of the first part of my talk. So I hope I have given you I've showed you some vignettes to suggest that articular cartilage is this really interesting and remarkable, soft tissue, which has, this interesting properties in the sense that it's, it has only, it's got very little regenerative capacity, but it's very tough and long-lived, right? And it can bear really, really large forces, right? With this minimal regenerative capacity, I showed you that the mechanical response of this tissue as a function of depth, we can understand how that changes using this rigidity percolation framework, right? And the fact that the shear modulus changes by several orders of magnitude as a function of tissue depth can be understood by using this, by understanding that the tissue is very close to this percolation phase transition, but at the same time, it's a multi-component material which is sort of buffering the mechanical response of the material a little bit, and it doesn't completely fall apart at this percolation transition. We also saw that if we want to understand crack propagation and crack resistance of this tissue, we really need to take into account the fact that it has this composite double network-like structure into account. And essentially what's happening here is the collagen network is responsible for the rigidity and the stiffness of the tissue, and its interaction with the flexible aggregate hyaluronic acid network is responsible for distribution and dissipation of stresses, giving rise to this enhanced crack resistance. And some of the things we are looking at right now basically is we are looking at what will happen when we take into account the fact that collagen is also a very hierarchical filamentous material, and in that networks in biological systems are not usually uncorrelated. You will usually often see correlated disorder, right? So if you take those things into account, how does that sort of change the kind of mechanical response that we are looking at? So this basically wraps up the first part, or the first story I wanted to tell you. And if you guys have any questions on this part, then I'm happy to take those questions now. Okay, should I move on?
I think so. Yeah, that sounds good.
Okay. Okay, now I'm completely switching gears, and the second type of tissue or tissue-like material I want to talk about is a co-culture of two different cell types. And the two different cell types, I'm going to focus on are the breast epithelial cells, MCF10A, and breast cancer cells, MDA MB231, right? And so, we are looking at a co-culture of these two different cell types. So co-cultures are really these nice experimental systems where you can grow, you can culture 2 or more different types of cells with different properties and of different types together. And then sort of you can study the self-assembly, the collective migration, and other collective properties of the systems in a controlled manner, right? So that's what we are focusing on here. And what we want to understand here is, so these 2 cell types that I'm going to tell you about, they have very different physical properties. So they have very different, Young's moduli, they have very different cell-cell adhesion, they have very different proliferation rates. And what we wanted to understand is how do the differences in these physical properties, sort of impact the phase separation, the self-assembly, and the collective motility of the system, right? And so these are all experiments. So the top panel are experiments done by my collaborators, Mingming Wu and Minglin Ma from Cornell University. And this panel is showing you how this, co-culture sort of self-organizes over timescales of days, right? And the second panel is showing data again for the same co-culture, but now it's showing you how this co-culture sort of self-organizes over, timescales of minutes and hours, right? So these are the same 2 cell types, the same type, same co-cultures, but the first panel is showing you what's happens over really large timescales. And I should mention that the cell division timescale here, so MDA MB231 divides over every 34 hours or so, and MCF10A divides over every 18 hours or so. So the healthy cells in this case actually divide faster than the corresponding cancer cells. So the top panel is basically data over timescales larger than cell division timescales, right? Or after you cross day 1, it's larger than cell division timescales. And here we are showing, you're looking at data that's a timescale smaller than cell division timescale. So that's sort of one way to differentiate these 2 set of observations. Okay, so let's look at what's going on in the first set of data. So we start with a co-culture of these 2 different cell types, and initial ratio is 1:1. So we have the same number of cells of each type, and they are seated in this microfluidic well, right? And you can see that initially they are randomly mixed together. That's the initial configuration. Now what happens at the end of day 1 is that these 2 different cell types sort of, self-organize in a way that you get this core shell morphology, where all the healthy green cells are on the inside and form the core, and all the cancer cells, the red cells, form the shell and are on the outside. And we can understand this self-organization by using the differential adhesion hypothesis, right? So what the differential adhesion hypothesis does is it models these different cell types as blobs of liquid with different surface tensions, and also there is an interfacial tension. So if you write down the free energy of this system and minimize this free energy, then this free energy minimum will correspond to this configuration which you see here, where all the sticky cells, the cells which are more adhesive, will be on the inside, and cells which are less adhesive will be on the outside. Now it's also known from experiments that epithelial healthy breast cells, they express this protein E-cadherin, which mediates cell-cell adhesion. So these cells have really strong cell-cell adhesion and form these confluent layers of cells, right? On the other hand, once these cells become cancerous and the more invasive they are, what happens is these adhesion proteins, E-cadherin expression is heavily downregulated, right? So this MDA-MB231, the cancer cells, are not that sticky anymore because they don't have as much cell-cell adhesion proteins as this MCF10A would have had. So the sticky cells, the green cells, will be then on the inside. They have more surface energy. The red cells would be on the outside. But then why does the core shell morphology reverse as you go to day 3 and then day 6 and day 9? It stays reversed. So now we have the healthy cells on the outside and the cancer cells on the inside, right? So we wanted to understand why and how is this, differential adhesion hypothesis getting modified as we go on and on and on, right? So to do this, we still, use the same free energy picture as before, as people use in differential addition hypothesis. But we now write down the energies for these two morphology, right? So morphology 1 is when the healthy cells, the green cells, are on the inside and the cancer cells are on the outside. And morphology 2 is when the cancer cells are on the inside and the healthy green cells are on the outside. And we can write down the energies of the two morphologies in terms of, the radii of, this green circle and the red, and then again this red and green, and in terms of the surface energies of the green cells and the red cells and the interfacial energy between the green and the red, cells. So we can write down this energy. We can also write down the difference between the energy of these two morphologies, the E1 minus E2, and we can sort of collect our terms in such a way that you now have this term you can express in terms of the ratio of the green cells and total number of cells, the red cells and the total number of cells, and the ratio of the differences between the surface tension of the surface energies of the two cells divided by the interfacial energy. So now at the end of day 1, cell division really hasn't done much. So this first term is still quite small, right? And so the second term is still dominating. And because the green cells are more sticky than the red cells, sigma G is larger than sigma R. So this second term is larger than the first term, and this negative sign means that morphology 1 has a lower energy than morphology 2. So we get this morphology 1. But as we have gone on to, day 3, day 6, day 9, now cell division has sort of taken over. And the number of cells for the green cells and the red cells are now very, very different. And remember, for these two cell lines, the green cells actually divide at a much faster rate than the red cells. So this term not only is quite significant, it can become larger than the second term. And when that is the case, this morphology 2 will now have a lower energy than morphology 1, and you will get this core shell reversal, right? So we can actually draw sort of a phase diagram in terms of beta and K, where beta is the ratio of the differences in the surface energy of the two cell types and the interfacial energy, and K is the ratio of the number of the two cell types. And you can see that if beta is sort of greater than 0.8 or so, then we will always get this morphology 1, which means that if the difference in surface tensions or surface energies between the two cell types is much, much larger than the surface energy is larger than this, then you will always get this morphology. But then you do have a region of phase space where you can get this other morphology where, you have the healthy cells on the outside, and the cancer cells on the inside, right? So essentially what's happening is cell division or cell proliferation or differential cell proliferation is changing the ratio of the number of cells, and that's sort of modifying the balance of these energies and giving rise to the second morphology. But this is still, the final state. So you might ask, okay, but how do we get to this morphology, right? So what does the initial phase separation of these systems look like? And to understand that, we have to look at what happens at much, much smaller time scales, right? So here I'm showing you experimental data from Joseph Case's group. And again, I have a co-culture of this breast epithelial cell, MCF10A, and the breast cancer cell, MDMB231. But now we are looking at what happens of the order of minutes and hours, right? You can see that starting with this sort of random mixture, the cells sort of start to form clusters, and then these clusters sort of start to coarsen. Now, what we wanted to understand is that is the evolution of the structure and the dynamics of this phase-separated co-culture similar to what you would see for binary liquids, given when we were using differential addition hypothesis, we were modeling the two cell types as liquid blobs, right? So can we keep using that analogy, and would the phase separation here look like what you would have expected in case of liquid-liquid phase separation of a binary liquid, right? So that is the next step. So to do that, we modeled the two different cell types as these active Brownian particles or self-propelled particles with different mechanics, so different cell-cell adhesion, because they express different extent of, the expression of equiadherin is different in the two cell types, but they also have different stiffnesses, because it has been seen now in many experiments that, if you measure the Young's modulus of this MCF10A, the healthy cells and MDMB231, the cancer cells, and no matter whether you're looking at the nuclear region or the cytoplasmic region, the cancer cells are always much more squishy than the healthy cells, right? So, and we wanted to take that mechanics into account in our modeling. So our model, again, has a self-propulsion contribution, and it has a mechanical interaction contribution. And the mechanical interaction comes into play only when the two cells are in contact, right? And when these cells are in contact, they can adhere to each other, which will depend on their Young's modulus, but also on their surface tensions, their surface energies. And if they push into each other too much, then there is going to be an elastic repulsion, right? Because they do have a finite Young's modulus. And this energy I'm using, or this force I'm using, goes by the name of Johnson-Kendall-Roberts contact force for elasto-adhesive contact, right? So before I show you the results for this co-culture, I did, my postdoc actually, did a control study for a system which has two types of cells which have the exact same Young's modulus and the exact same surface energy, but they know that they are different from each other in the sense that there is an interfacial energy, right? And if I have a system like that, you can see the system phase separate, and then the clusters, the phase separated domains, will coarsen as a function of time. And if we want to understand the phase ordering of a system like this, sorry. Yes, if we want to understand the coarsening of a system like this, we look at how, we write down the density structure factor for these systems, in this. So it will follow this kind of a scaling relationship where basically L is the dominant length scale of the system, and D is the dimensionality of the system, and G is a scaling function which decays as a power law, and then theta is basically the power for this power law. So if we have clusters that are very compact and have very sharp interfaces, then this power law will have a power, theta will be D plus 1. So in 2D, this should just be 3. And indeed, if in our simulations where the two cell types were exactly of the same type, we do get this power law, this power to be 3. The dominant length scale, on the other hand, so it will grow, with time, again as a power law. And this power law, we find this to be 1/4. Now, those of you, who are thinking of, sort of like an Oswald ripening kind of phase ordering, you would have expected this to be 1/3 and not 1/4. The difference between that situation and this situation is the following. So in both cases, sort of the merging and coalescence of clusters is driven by diffusion. But in that case, the diffusion is thermal diffusion. In this case, we do not have thermal diffusion, we have rotational diffusion. And so the diffusion constant has a different dependence on this length scale. And as a result, we have the scaling, the dominant length scales goes as T to the 1/4 and not as T to the 1/3. Well, so this is our control case. Now let us look at what happens in our actual case, right? Where we have these two different cell types with different Young's moduli and different surface energies. And their self-propulsion speeds are exactly the same. So once again, we can see that they phase separate and they form these clusters, but now the healthy cells form these very compact clusters, and the cancer cells form this much more dispersed clusters, right? And the boundaries between the two clusters are not sharp anymore. So then the scaling for the density structure factor is no longer -3. So the -3 scaling also has a name. It's called Porod's law. But our scaling now shows this deviation from Porod's law because the boundary is no longer a sharp boundary. And this deviation is even more strong or more pronounced for these cancer cell clusters, because not only is the boundary not sharp, but the clusters themselves are also not compact at all. And to look at this in a little bit more detail, we also looked at the distribution of cluster sizes. And what we found is that for the cancer cells in particular, there is a hierarchy of cluster sizes, and we can sort of collapse the cluster sizes onto a power law, right? And that sort of is suggesting that, you know, we get this very dispersed, rough clusters for cancer cells, and also there is this sort of hierarchy of sizes of these clusters, which is basically giving rise to this departure from the -3 scaling. We also looked at the collective movement of these cells. So we looked at the trajectories of the two cell types, and we can see that the healthy cells are mostly confined in their regions and do not move very much. The cancer cells, however, have these bursts of motion, right? And this enhanced motility of the cancer cells is also reflected in the mean square displacement, where the mean square displacement of the cancer cells is always larger than that of these healthy cells. And please, I want you to note that the two cell types have exactly the same self-propulsion speed, right? So the enhanced motility of the cancer cell is arising from the differences in the mechanical properties between the cancer cells and the healthy cells, right? And this might help to, you know, sort of explain these experiments by the Wirtz group, where they saw that if you have a cancer cell surrounded by other cancer cells, it moves a little bit, but not very much. But if you have a cancer cell which is tagged, it's surrounded by healthy cells, then the differences, and again, these are the same two cell lines, MCF10A and MDMB231, then the differences between the mechanics of the two cell types can sort of help, you know, lead to this enhanced motility of these cancer cells. So that wraps up the second part of my talk as well. So to complete, I guess, to wrap up everything, I showed you how collective behavior sort of can give rise to very interesting mechanobiology for two different types of tissues or tissue-like materials. We first looked at cartilage tissue or articular cartilage tissue, and we saw that how this rigidity percolation transition sort of can help the tissue tune its mechanical response by several orders of magnitude as we go along the depth of the tissue. We also saw that the toughness and crack resistance of cartilage tissue can be explained by thinking of the tissue as a composite double network. And then I told you about a completely different system, which is a co-culture of two different cell types where the mechanical response is coming from the cells, and we are really not worrying about the ECM at all. And in this case, we found that over time scales that are much larger than the cell division time scale, the sort of self-assembly and collective mechanobiology is mainly governed by, you know, the interfacial tension and the surface tension of the cell types, as well as the rates at which the cells are dividing at time scales smaller than cell division time scales, where cell division doesn't really matter very much. It seems that in addition to the adhesion properties, the mechanics, the mechanical stiffness of the cells can also play a very important role, and in fact can give rise to this basically phase separation, which has different scalings than what you would have expected for scalings for just binary liquids. With that, I guess I would thank all of you for coming to my talk, and my students, my collaborators, and my funding sources. And the names that are in red are is everybody who has contributed to this work. So with that, I would end and take any questions you have. Thank you.
Great, thank you, Moumita.
Go ahead, John.
Does anyone have any questions?
I just have a quick one. Is there any physical meaning to the sort of surface tension between the two different cell types?
Yeah, so basically, you can sort of correlate the surface tension and the interfacial tension to the adhesion, right? So when you have more sticky, so when the cells are more sticky and they have more adhesion, you know, if you think of these cells as liquid-like, that will correspond to a larger surface tension. So that's sort of the map.
Okay, thanks.
I have a question going all the way back to part 1.
Mm-hmm.
I guess maybe it's more on the experimental side. You know, like what can Itai and his lab do with the collagen hyaluronic acid aggrecan complex? Like, is there any way to, you know, bind more aggrecan or, you know, change the sort of mechanics of this
So
continuous network to kind of justify your predictions from your models?
Yeah, so some of the things that we are trying out right now is you can use different types of enzymes that will digest different types of these biopolymers, right? So,
Yeah.
you know, so you can use collagenase or trypsin or some other enzyme that will preferentially digest collagen or digest, you know, one of the other biopolymers. And that way you can change the concentration of these, you know, different types. You can completely even get rid of, let's say, the aggrecan, right? And, you know, you can sort of see how well then the mechanical response that you get, you know, will agree with, you know, the models that we have come up with, right? And one of the things that we are seeing, the thing I did not talk about, is that qualitatively the mechanical response agrees with what is predicted in the model, but the percolation threshold is shifted a little. And that's why we are now looking at, you know, correlated networks, because if you have correlated networks, then that can give rise to, you know, for certain degrees of correlation, you can get a lower percolation threshold with the same amount of filamentous material.
Sounds good. I guess I have a very similar question for the second part too, which is, I guess these cells are contained in these things that keep them separated, seemingly. Can you modify the surface interactions there too to sort of play the same tricks?
The second part is much harder, right? Because, but again, you can, I guess you can modify, right? Because, you know, the wild type cells have certain, you know, they will express certain proteins in a certain manner, but maybe you can chemically treat them so that, you know, you can chemically sort of perturb or depress the expression of some adhesion proteins, right? But those experiments are more difficult to control because the way they are done is you make this microfluidic spheres or microfluidic cylinders, and you then put your cells in these cylinders, and then you just let them do whatever they want to do, right? So, you know, so the amount of control you have over them is a little bit less, but I guess you can maybe still inject, you know, your different chemicals that will probably help to change them. But these are more difficult, I would think, or I think.
Hi, Moumita. Seth Fraden speaking. In your first part, in your model, you had this coupling between the soft network and the more brittle network. Yeah, I guess that E3 term. So can you just, is that the coupling is E3?
Yeah, the coupling is E3. And I think one of the things I want to point out is the coupling is not at the nodes, right? It's basically, think of two networks, and, you know, there is some weak interaction between the midpoint of, you know, bonds in one network with corresponding bonds in the other network, and the networks are staggered a little.
Okay, and so those X's are the bonds,
not interaction at the nodes.
And okay, so here the X's are the bonds, and then you just you want them to be parallel to each other.
We want them, it's a little bit staggered, but there is interaction between bonds in the two networks. And one of the other things we do is, like, you know, so if either of the bonds is missing, then that connection is also missing, right? So for the connection to be there, both the bonds, like in the corresponding networks, have to be there. So that's sort of how the model is constructed.
Okay.
But you can also do other kinds of situations, which we are looking into now, where one network is rigidly cross-linked, and the other network may have some combination of cross-links and entanglement by, you know, making the network more like a phantom network.
And are the networks the same length in some sense?
So that's, as of now, they have the same rest length, but yes, if you want to do it more accurately, then, you know, the hyaluronic acid proteoglycan network, that's not just more flexible and more floppy, but the rest lengths and the, you know, the cross-linking distance there should also be very, very different.
Yeah,
so that's not there in the first iteration of the model, because this was more of a proof of concept model, right?
Yeah,
when we get this kind of fracture behavior. But in the next iteration of the model, we do want to incorporate that.
And then you anticipate any qualitative differences if you go to 3D rather than 2D?
I think I think in terms of the fracture behavior, we don't expect qualitative differences, but you know, the ultimate tensile threshold and other things will shift quite a bit. And the other thing I want to point out here, you know, cartilage it's a three-dimensional material, and not just that, there are a couple of different things. I showed you that the organization of collagen fibers can be different depending on depth. There is that. Also, there are three zones in the tissue. There is the superficial zone where, you know, the networks are very sparse. There is the mid zone where, you know, they have intermediate density. And then there is this deep zone where everything is very dense, right? So that is probably going to have, like, qualitatively, there may be a big effect, but that's because of not just the fact that you have like a double composite network in 3D, but also because in 3D, you have this gradient, you know, in the density of the fibers and also the orientation of the collagen fibers. So when we do 3D, we would want to take those things into account, and those probably will make this not just much more complex, but also, you know, much more interesting.
Thank you.
Is there anyone else that would like to ask a question? It seems not. All right. Well, with that, I think, you know, we should thank Moumita one more time for the very interesting work and the great presentation.
Thank you. I wanted to make an announcement before I leave. Two announcements. One announcement is that I have a postdoc position in my group, which is completely different from this. It's about construction of a single synthetic neuron and a network of synthetic neurons, and, you know, connecting their structure to their function, how they do computation, but not in biological neurons, synthetic neurons, right? So if any of you are interested in, you know, structure function properties of networks, and not just mechanical properties, but also, you know, in this case, it will be electrophysiology, those kind of properties, right? So it's still percolation, but different type, then please get in touch with me. The other announcement I have is I am a member of the DBio Executive Committee, and proposals for focus sessions and invited sessions are due on June 1. So if you have interesting ideas for focus sessions or you know, invited sessions that you want to host for, you know, the next upcoming March meeting, please submit your sessions.
Where is the next March meeting?
March 2021.
No, where?
Nashville.
Okay.
Yeah.
Excellent.
Mm-hmm.
Well, we're looking forward to that.
Yeah, thank you again, both Seth and John. It was a pleasure. And,
thank you for participating.
Yeah, and I actually look forward to participating in the other coming talks, and I've asked my group to do that as well.
Well, very welcome.
Great.
Thank you.
Thank you.
Take care.
Bye-bye.
Bye.
Thank you.